The NIH-funded Antimicrobial Resistant Leadership Group. ARLG is changing the field of clinical research in antimicrobial resistance, and today we will talk with the principal investigators of ARLG to discuss their strategy. We will try to review the history and accomplishments of ARLG, discuss the missions of ARLG, including training new clinical investigators focused on antimicrobial resistance, and comment on the future of ARLG and clinical research on antibiotic resistance. Welcome to Editors in Conversation. This episode is brought to you by Antimicrobial Agents and Chemotherapy, available at aac.asm.org. I'm your host, Cesar Arias, Editor-in-Chief of AAC, and this podcast is supported by the American Society for Microbiology, which publishes AAC. Don't forget to check our latest December issue of AAC with really outstanding papers on resistance, including several papers related to SARS-CoV-2. Joining me to talk about ARLG are the principal investigators of this ambitious effort. Very distinguished people here. Dr. Vance Fowler, who is Professor of Medicine and the Florence McAlister Distinguished Professor of Medicine and Professor in Molecular Genetics and Microbiology at Duke University. And Dr. Henry Chip Chambers, Professor of Medicine Emeritus of the University of California, San Francisco. Welcome to Editors in Conversation, gentlemen.
Thank you.
Thank you. Good to be here.
So I understand that. So ARLG has been going on for more than now 8 years, Dr. Fowler. So many people may not be aware with actually how ARLG started and its history. So I think it's very important to put people into context. Can you comment about how you started this and how where we are right now?
Sure. Thanks, Dr. Arias, and I appreciate the opportunity to talk about the ARLG today. The ARLG started, or ARLG, or Antibacterial Resistance Leadership Group, started in 2013 and was an initiative that began with an RFA from the NIH to address the growing concern of antibacterial resistance in the scientific community. The overarching mission of the Antibacterial Resistance Leadership Group is to identify, prioritize, design, and execute clinical research in antibacterial resistance. And so with that mission in mind, Dr. Chambers and I were able to collaborate to begin a submission that was ultimately successful and initiated in late 2013 or early 2014. And from that time until now, we've really had the singular privilege and honor of being able to work with some of the most talented investigators anywhere, in my opinion at least. To focus on antibacterial resistance. Time's gone by, and during the first round of the initiative, which lasted about 6.5 years, we ultimately initiated over 40 different studies that involved over 21,000 subjects in some fashion or another. There were 130 sites in 12 different countries. And ultimately producing over 130 publications, and 2 new FDA indications in the setting of diagnostics. So, boy, what a whirlwind, but man, it's been a lot of fun.
So can you, before I go to Dr. Chambers, about the monetary investment on the NIH, so how much money has taken to do all these studies?
Sure. So the initial round, which again was about 6.5 years, was a little over $65 million of total dollars. That's directs plus indirects in terms of the overall investment. Now we've just had a competitive renewal, and we were fortunate to be able to continue our efforts in ARLG 2.0, which we affectionately refer to it. And in that, this new round, which began in December, so a little less than a year ago, there was about 100 million total NIH dollars invested.
Fantastic. Dr. Chambers, can you explain the audience how the structure of ARLG is and the different components and the goals of these components to change the clinical research arena in antibiotic resistance?
Yes, certainly. And thanks, Dr. Arias, for having us on this podcast and the opportunity to share more about ARLG. I'll start at the topmost interaction, and it starts with NIH and NIAID, the National Institutes of Allergy and Infectious Disease, and specifically DMID, the Division of Microbiology and Infectious Disease. There are collaborators and colleagues in this effort. NIH, as you know, funded this, and we work very closely with them. So with respect to ARLG, the leadership of DMID and ARLG meets on a regular basis and on an ongoing basis as well through a variety of committees and subcommittees. So for the ARLG itself, I'll start at the decision-making body and the body that approves all the research protocols and sets the final agenda and establishes priorities, and that is the executive committee. Dr. Fowler is chair of the executive committee, and I am the vice chair. There is representation from the research community as well, from some of the other committees that I will explain in a moment, and from the leadership of DMID. The other important group within ARLG, which reports to the executive committee, although it's really much more interactive than that. You're used to seeing arrows going up and down and sideways, and this is really a reciprocating arrangement between the executive committee and the subcommittee, although administratively the subcommittee reports to the executive committee. The subcommittee, the steering committee, not the subcommittee, the steering committee recommends to the executive committee priorities and agenda and specific studies. Those studies are reviewed by the steering committee, vetted and approved or not, and then referred to the executive committee for final action. The steering committee also is the body for discussion of new protocols, for review of protocols outside the ARLG, which we also are interested in seeing, and those that are generated within the ARLG. They are important for recommending governance or changes, and is really the day-to-day working body that interacts with the scientific subcommittees, which is then the next level. The scientific subcommittees are in three areas: diagnostics, gram-positive, and gram-negative, focusing on resistance primarily, but trying to advance the field of diagnostics, which is so key with dealing with resistance. And each of those subcommittees considers proposals, recommends them to the steering committee, and also reviews any outside proposals. I should also mention that there are two other important committees that report. They interact with the steering committee, but they really report directly to the executive committee because we think it's primary importance, and that is an innovations committee headed by Dr. Boucher, which considers innovative approaches to approval and design of clinical trials. And then the Diversity and Mentoring Committee, which is a huge effort by ARLG to make sure that we're training. When Vance and I ride off into the West, there will be some crew left behind to continue the agenda. So we offer a fellowship, seed grants, and a program called Trialists and Training. Vance, you might want to elaborate on that, but I think I've hit the high points. I want to stress that ultimately, this is an effort that at the highest level is between ARLG leadership and ARLG itself, represented by Vance and me, as well as some others, and DMID.
Thank you very much for this thorough explanation. We are very, and I'm very, very passionate about training, Vance. And I know one of the missions from the inception of ARLG was to try to find mechanism to train the new investigators. We have a very good, big audience of younger investigators for this podcast and for our journal. So what is the ARLG fellowship, and what is the training mission of ARLG to try to stimulate these young investigators to join ARLG?
Yeah, glad you asked that question, Cesar. The next generation is so important in so many ways, and I'd say with antibacterial resistance, it's particularly important just because this problem didn't go anywhere. We've got our hands full, all of us, in terms of combating this challenge. So the next generation of investigators is vital. In the first round, I'm honestly very proud of what the ARLG was able to do in the area of mentoring. The first round, it was primarily under the leadership of Anthony Harris at University of Maryland. And there were a total of 40 mentees in some way or another that were involved in 25 different ARLG studies in the first round. And we hope to do as well or better this time. Basically, one of the first key sort of realizations about mentoring was, particularly in general, but for antibacterial resistance in particular, is that different stages of trainees need different types of support. For example, a junior faculty who is investing in a particular project, he or she will have a whole different set of demands and realities and resources than, for example, a medical student who may have a limited period of time, and it may, in which case the journey is the destination. So because different trainees need different things, we've tried to develop programs that can reflect those realities, and there are three sort of basic programs that we pursue. One of them is called the ARLG Fellowship, which we'll talk about a little in detail later, but to my view is just a kind of a Cadillac version of a fellowship training, in that it provides 2 years of full salary support and tuition support for formal didactic training and clinical research. So something that really, personally, I'm very passionate about as a resource. There's a second program called the Early Stage Investigator, or ESI Seed Grants, and this is a program in which you apply to get up to $50,000 of direct support for a first project. And this we designed this project with the intent that you could kind of put the training wheels on the bicycle, if you will, a little bit for that first project. That it got enough funds to get out there and get some preliminary data, maybe get a paper or two that sets them up for that for a more definitive career K-type series of grant. And then the third one was, honestly, was sort of a we sort of learned as we went along, and this was what we call the trialist in training. Trialist in training is typically targeted towards the junior faculty who is an instructor or assistant professor type role, and who is ready to go all in on clinical trials and drug-resistant bacteria. And in that instance, what it allows is that individual is afforded the opportunity to essentially dive into a project, one of the ARLG studies, and learn by doing. That is, interacting with the study personnel, the things that that physicians don't generally think about when we're talking about trials, but ultimately are the things that get it done. And so ARLG Fellowship, ESI Seed Grant, and Trialist and Training are kind of the three levels of our mentoring. And I'll stop there.
Thank you very much for that. I think these are tremendous opportunities for all our trainees and young talent that are going to be listening to this podcast. In the same vein, Chip, just getting into the matter of ARLG has contributed. Can you comment about the most impactful projects you think happen, at least in the first phase of ARLG, that are setting up the agenda of clinical research, not only in the United States, but globally?
Yes, I would highlight 3 or 4. It'll be interesting to see if Vance would agree with these. And these are in no particular order of priority. That's just as they come to mind. But I'm going to start with gram-positives because, as we know, Staph aureus is one of the most important drug-resistant pathogens.
And very interesting. Very interesting.
Yeah. And then we'll get to the gram-negatives. But, of course, Staph aureus is really deserves a lot of attention and interest. So one of the first studies we did is a study called PROVIDE. And PROVIDE was a study to examine the relationship between parameters of therapeutic drug monitoring for vancomycin dosing in patients with staph bacteremia in terms of outcome. And we focused on AUC, area under the curve, as a predictor of outcome and asked the question, what is the optimal dosing regimen to achieve an AUC as a target, and how does that affect outcome? And what it showed is that there was an optimal target. It was 515, more or less, AUC of micrograms per mil times hour is the units. And that going above or even a little bit below that did not substantially affect outcome. In fact, it came at some cost. And those observations, it was an observational study with all comers, so not a randomized control trial, but an observational study that was very robust, prospective, predefined, central laboratory, a high quality study run by Tom Lodise that showed that higher AUCs really don't buy you any benefit, are more toxic, and in fact are associated with somewhat worse clinical outcomes too in terms of cure and mortality. And those findings led directly to the current guidelines for vancomycin dosing from IDSA and the American Society of Hospital Pharmacists. So PROVIDE. Slightly in a different area is the Radical series, which continues. This is really a program of several studies that are ongoing, run by Ephraim Salek, that attempts to define an approach to testing, if you will, that's based on host genome response or host gene response to infectious diseases to differentiate viral from bacterial causes. Sort of a more refined procalcitonin, if you will, and it looks like it performs at least as good as and probably better than procalcitonin. And that has led to a series of investigations and in fact received an award as a diagnostic to assist in the diagnosis of respiratory tract infections. Why are we interested in that infection? It's one of the most common reasons that antibiotics are used, and it turns out one of the most common reasons why antibiotics are abused. So Radical, which continues today, a series of studies that looked at diagnostics in gram-negatives and gram-positives to evaluate the utility of rapid diagnostics, and a study called SCOUT-CAP, which looks at duration of therapy in children with pneumonia, 5 days versus 10 days. This study was just completed recently, and the high-level data suggests that, in fact, I think shows that shorter course of therapy are probably better than the standard 10-day course. I think that will also change guidelines. There's a microbiomic component of that study, which is going to be very informative about the effect on the microbiome of children who receive longer course of therapy. That study employed a novel study design, which is the last thing I'm going to end upon, is we've been exploring new ways of doing clinical trials that are more informative, that allow a superiority design in clinical trials, as opposed to the traditional non-inferiority trial. It's called DOOR, and it uses an ordinal scale, which we've kind of become familiar with COVID and COVID clinical trials, but tuned for bacterial infections, and it's desirability of ordinal outcome ranking is what DOOR stands for, and it allows you to address outcomes in a way that is more global, and it recognizes that not all outcomes are the same, and neither are all cures, and there are better cures and worse cures, and better outcomes and worse outcomes, and it's not simply thumbs up, thumbs down on outcome, and that we should be including these other considerations in terms of the patient's journey to an eventual success or not, but scoring that in a way that is more informative than the traditional clinical trial design. I'm sure I've forgotten something. Those are my favorites. Vance, what did I forget?
Well, the problem with asking, asking us about this is it's sort of like asking.
Oh, I know what I forgot. MDRO.
Thank you very much.
How could I forget MDRO? Well, because it's gram-negative, right? So MDR is multiple drug-resistant organism. It's a network that started out, I think, in David Van Dyne's basement or something. I don't know. He's at North Carolina and was interested in carbapenem-resistant gram-negatives, and had a number of centers that were selected. Was kind of more of an idea than really something put in full-on execution, and approached ARLG to see about expanding knowledge in the area of carbapenem resistance in enteric organisms, and it focused on the enterics, primarily E. coli and Klebsiella, a few others, and asked the question is, well, what is the clinical landscape? How do we best collect data? How can we use that information to inform clinical trials? What's the molecular epidemiology across the United States? What is the impact of this organism in the US? And so that got expanded across the United States, and then, Cesar, as you know, eventually internationally, and included sites in Colombia, other sites in South America, China, Southeast Asia, and broadened really its footprint into what started out as Crackle, and I can't remember what Crackle stands for, but somewhere there's some carbapenem resistant organisms embedded in there, and became Crackle 2 as an international lead on from Crackle, which then has grown into the MDRO network, which aspires to eventually take on the problem of drug resistance, primarily more drug resistant organisms, on an international scale, and to conduct clinical trials in that arena. So yeah, actually, we're talking about gram positives, but MDRO is in gram negatives. I mean, I think we both have to admit that the real challenge facing us is resistance in gram negatives in terms of therapeutics. That is an area of tremendous medical need. And I'm sorry I forgot it, but I eventually remembered.
Vance, any comments about that? And you can comment about that, the global footprint of ARLG now, US and international.
Yeah, sure. Well, I mean, it's kind of like asking a parent, which of their kids' projects is their favorite. The little porcelain dog, or the little footprint, or the little handprint that they print. I mean, there are just so many cool projects with this ARLG thing. It has just been so much fun, and learning so much. I do think the Crackle is, an MDR has been a big, a big plus. It's funny that I will tell you how it started. It started literally with David Van Dyne and I sitting down at a coffee shop before COVID, talking about ideas. And he had created this network in Ohio, and sort of looking at CRE. And he said, well, we had done this some years before. We had done something along these lines with endocarditis, a thing called the International Collaboration of Endocarditis. And we said, well, why don't you, put some in to ARLG, and about expanding that platform across the U.S. And then let's see how it goes, and maybe we can do it internationally. And the first international call, and Cesar knows as well, was essentially to link David Van Dyne with Cesar. And the rest is history. Then the NIH brought in, they had some collaborators or investigators in China, and 3 or 4 trips to China later, they're enrolling, I don't know, something like huge numbers of cases. I think it really is, what is it going to allow us to do? You could say, oh, it's an observational cohort study. What in the world does that do? But here's what it does. It confuses the issue with facts. It's if you really want to treat an infection, step one is you need to understand the infection. And I think probably the biggest lesson, that to me at least, has been that this infection that we're calling one and the same and treating one and the same is actually pretty different geographically. It has different mechanisms of this resistance. You got to get into the molecular elements of why you've got a resistance of interest. It has different implications in terms of the sites of infections. There are even different cultural aspects that enter into what samples are obtained. For example, if particular cultures are less inclined to obtain blood cultures, but more inclined to take sputum. So all of these things factor into it. And I at least had no clue about all that until we began to actually look at it carefully. So why is that important? Well, it's important because now, once you confuse the issue with facts, you're actually able to design a clinical trial that can deal with this in a robust manner and get to the intervention, which is what all of us care about.
Yeah. So Chip, this is a fantastic view on this. So in this current, what Dr. Fowler calls 2.0, so what are the main goals of this stage of ARLG?
So as we started with very little or nothing except some ideas, and just by the nature of designing a clinical trial, it's easier to do an observational study. So much of our, I guess, proposals that were actionable and feasible were observational in nature. And over the period of months to years, we began to be able to design interventional trials. And 2.0 expands upon that and is more ambitious in terms of designing interventional trials. We have targeted gram negatives in a very important way and are designing a trial to address the problem of lack of oral therapy in patients with ESBL, extended spectrum beta lactamase, producing gram negatives, and hopefully to improve treatment in that area. And we chose that pathogen really based upon feedback from the community of a tremendous need. So more interventional studies, more studies that will give high quality data in interventional trials. There is a study of Staph aureus bacteremia, for example, that will look at the utility of, I don't know if it's step down or step over or what, but of dalbavancin to complete a course of 6 weeks of therapy for complicated Staph aureus bacteremia, allowing patients to be discharged and treated with a couple of doses a week apart of dalbavancin. The preliminary data, again, observational, look pretty good. So we've designed a trial to compare this in a randomized manner, which will give us high quality results. It also employs the DOR analysis, so it will inform in a more granular level the benefits of this type of therapy over traditional standard of care. I've mentioned the radical study, which is another iteration and moving towards identifying a platform, which I think will eventually lead to an FDA approval of this as a diagnostic test. We've expanded into the international setting, building a bit upon the MDRO network of using diagnostics to improve outcome in drug-resistant gram-negative infections. And just to put in a plug for the international efforts, you really can't do a drug resistance trial very readily in the United States. It's an international problem, and much worse in many places outside the United States than inside the United States. So recognizing this and where the need is and where the studies can be done, we have moved more into the international realm as well in conducting these drug-resistant studies.
Yeah. Vance, based on that, so I know there are other international networks like in Europe and maybe Australia and others. So what is the connection of ARLG with those networks? Are there any agreements reached or in process to try to expand the reach of all these studies to be meaningful across the globe?
Sure, yeah. And the short answer is yes, there are a number of them. And the nature of the relationships are, vary according to the particular projects in place. With regards to the MDRO network, the contracts are primarily directly to the centers. Certainly, as within places like China and parts of the Pacific Rim, South America, and others, they're directly between Duke and the center of interest. That helps for the purposes of observational studies. More broadly, we've got relationships in place with Combact in Europe, and there's been some bi-directional initiatives through that arrangement to where sort of not directly with ARLG, but through some of the partners of ARLG to undertake studies such as vaccine, setting up work for vaccines for E. coli, and understanding HAP-VAP trials or HAP-VAP populations in European ICUs. Have been some of the work that have gone back and forth, sort of in parallel to ARLG. I think we're always looking for good partners to try to advance the field. At the end of the day, there's a finite amount of funds and there's a finite amount of time, but there's also so many really great investigators out there that we'd love to partner with that hasn't happened yet. So we're open to always open to that.
Great. Chip, I am biased, but the molecular epidemiology and laboratory-based susceptibility testing and how the field is changing right now, that if we have some advances now to combat resistance is probably related to diagnostics and the way to implement rapid diagnostics and other technologies that are really coming to fruition very rapidly. So can you explain what the laboratory component of ARLG is, and how is that envisaged to be developed as an important tool for resistance?
Yes. So the laboratory center is run by Robin Patel. She leads it. And they have their own initiatives, which I will talk about in a minute. And I think you're referring to GenoSmart as probably the most innovative. They participate in every study that we design to inform the laboratory components of every trial. I'm going to give an example of a study that we didn't talk about, but it's called PHAGE. And this is a phase 1, phase 2 study to look at the efficacy of phage therapy in patients who have cystic fibrosis and are stably colonized with Pseudomonas. And the question is very simple, and it's not been answered, is what is the microbiologic effect of phage on the target organism in patients who have high burdens of infection or colonization in this particular case? And in designing this study, we found out that, well, nobody really knew how to measure the impact microbiologically. How do you do pharmacokinetics of phage? How do you monitor phage in the bloodstream of the target organism? How do you test for resistance and susceptibility in a meaningful manner? And that has just brought up a number of needs to improve trial design and what very basic questions in developing an antibiotic. And we're approaching it from that point of view. And the lab center is taking a lead on that and helping us go through that. The other initiative is, and I think you're right, the field, actually the technology has gotten way ahead of the application of, and it's been pretty rudimentary, really. It's been PCR based, a limited number of targets. And I think, I know Cesar agrees, in a few years, we're going to be using whole genome sequencing. And how do you employ that technology and make it accessible to the clinician? You know, if you think there is confusion over susceptible, intermediate, and resistant on phenotypic testing, just take a look at how to interpret genomic information. And so GenoSmart is an effort to merge information about drug resistance phenotypes with some clinical data and to assist in predicting based upon high quality, deep molecular information for drug susceptibility. Because, you know, if you're doing MICs, it's 2 to 3 days down the line. We have a patient who has what I'm pretty sure is MRSA, endocarditis, and major and meningitis, by the way. And it's day 3, and we still don't know if this is a resistant organism or not. And I kind of wish I knew what we were dealing with. And the gram-negative field is even worse. At least there is a PCR for McA that can be useful once you get the organism isolated. But if you can just load a sample into a sequencer and do next-gen sequencing and get high-quality reads, you know, in principle, if we can get the informatics figured out, we're talking about a day kind of thing. So we're trying to explore if that's going to be possible or not. I don't think we'll know how applicable it is without doing studies like this. And it's going to take a while to figure it out, but we've got boots on the ground to try and do that.
Vance, any comments on this?
Well, I think that, you know, the diagnostic part is the future. And I am really happy with the way ARLG has contributed to that future. You know, we in the clinical realm, I think we're all familiar with the crisis that is facing the anti-infective development in terms of antibiotics. What I didn't know before I started this gig was that the crisis faced by diagnostic companies is at least as bad and sometimes a lot worse, just in terms of being able to implement and roll it out and maintain feasible products. So, one of the things that we've tried to do is to support diagnostic companies or provide some resources that can assist that with by way of some biorepositories that give them access to well-characterized isolates that they that can then use for their diagnostic purposes. We provided consultation on a number of these platforms. And I probably one of the things that I'm most excited about in our diagnostics area is some of the ideas. I mean, at this stage of life, honestly, ideas are what get me most excited. And a really cool idea that came up a couple of years ago, honestly, came up with a conference that NIH set up was a thing that ultimately has come to be called the Mastermind program. Whereby, and the idea is really simple. It just came out of the lab. I know as both of you have labs, we'd often use the same samples from one patient, sera or what have you, in multiple different experiments. And one of the questions became, why can't we enroll one patient to simultaneously inform more than one diagnostic platform, right? That what it does is obviously cut the cost of the trials dramatically, and makes it feasible. And so, long story short, we did that with 2,500 patients and 2 platforms for extragenital GC. There had been no FDA indication approval for that. We worked collaboratively with the FDA to develop a reference standard, against which to compare, call it a gold standard, if you will, and then partnered with different diagnostic companies and ultimately got FDA approval through one study for 2 different platforms. Well, see, now that's an idea that you can roll out in many other different settings, and something we'd like to do moving forward.
Great. I just want to spend the last few minutes of the podcast thinking about my ID fellows who are interested in antimicrobial resistance. And then say, how do I get on this? This all looks wonderful, ambitious, sounds like, a dream here. How do they get into this? And I know the ARLG fellowship is something very important. So Vance and Chip, why don't you tell them what to do for this fellowship and how they can apply in all of this?
Chip, you want to go first?
Yeah, I'll take a shot. First of all, it's not just the fellowship. I'll let Vance talk about that, but I want to encourage people to think more about the seed grants, the early stage investigator grants. It's easy to put in an application for it. What you need is a good mentor who is interested in your career and the project. You need a good idea. You need some guidance in how to write a proposal to get funding, but anybody can apply for that. And one of the a major emphasis for the coming cycle of funding is, we've learned through COVID about how infectious diseases disproportionately affect certain populations. So one of the things we want to take on, and it turns out drug resistance is very similar. For example, with MRSA, it disproportionately reflects minority groups. So we're interested in exploring that a bit. So I think if you have a great idea on how to advance the field, either because of your personal interest or because of a certain need, we would love to see those applications in an ESI grant. You can go to the ARLG website and it tells you how to do it. I think the track record on that's certainly better than an R01 or a K. I think it's around a quarter. Isn't that right, Vance? About 25% of those get funded.
Better my R01.
Yeah. And then I'll turn it over to Vance to talk about the ARLG fellowship.
Yeah, thanks, Chip. I mean, yeah, I think for the trainees, check us out, arlg.org. There are options there and there are resources there from the perspective of the ESIC grant and the fellows. There's the fellowship. What do you need? Chip hit it. I think step one is you need a good mentor. I think just like most things professionally, it starts with having a good mentor, and this is no exception. From the perspective of the ESIC grant, we have sort of a rolling admission. You put it in, it's going to be reviewed. It's actually reviewed in both in from a scientific perspective and through our mentoring subgroup. So we're interested in that. We'd like it to be focused in some way on the clinical end of the spectrum. So preclinical things will probably not be quite as responsive. With regards to the ARLG fellowship, there's generally one or two of those a year. I think it's a pretty big deal personally. As I mentioned a little earlier, it allows or provides for a trainee to have 2 years of research funded, salaries covered. And they can they can have support. It provides support for getting an additional degree, formal didactic training, leading to a master's or what have you in clinical research. It's in many ways, I think it sets people up for a K award, and I'd say our track record of persons who are ARLG fellows who went on to get Ks was, I'm pretty proud of what those numbers look like. So
80%.
Yeah, there you go. Well, so thank you for considering that, and we'd look forward to receiving competitive awards.
Well, I want to thank you both for this amazing conversation. This is so exciting. I just want to invite everybody that we are going to have our annual Texas Medical Center Conference on Antimicrobial Resistance and Stewardship in collaboration with ARLG. The blog, the website is amr2021.blogs.rice.edu. And ARLG will have the opportunity to present fantastic projects that we commented here. We also have a session for early stage investigators. The conference also has mentoring breakfast for everybody, including to try to stimulate women and minorities into this field. So it's a 3 days that is going to be taken. It's going to be held in January 26, 28, virtually, and in 2021. Any final thoughts, Dr. Chambers and Dr. Fowler?
Great opportunity. We have 6 more years to make an impact, and I think by that time we'll have 3 or 4 studies completed that will be high quality, and I think change how we treat and diagnose infectious diseases. It'd be good if we had $1 billion, maybe 3.0. We can get closer to that, but we're happy with what we have now, and that's where I think we're headed.
Vance?
It's been a thrill. It's that this the one thing about this AMR is just how diverse the problem is. Different bacteria, different disease states, different diagnostics, different, there are cultural issues, there are diagnostic issues, there are therapeutic issues. It is never dull. And we were always interested in, promising people and exciting, joining this big struggle. So thank you for the opportunity.
Thank you, gentlemen. This is Cesar Arias, Editor-in-Chief of AAC, signing off.